Recerca Matemàtica that are very welcome here, understand a little bit what we do and how we are organized and the kind of research we do, okay? And then I will just summarize a little bit the contributions from the ICM that will be presented today, tomorrow, and the next week that we will be at the campus in Autónoma, at the CRM premises. So here, all of you know, but if you want the program, just take this QR code and you will see. So first of all, we are the Institute of Marine Sciences. We are Severo Ochoa since 3 years ago, and we belong 100% to CSIC. So we are not a mixed center or a combined center. We don't have any kind of complexity in this sense in our organization, and we belong to the ICM. But we are a center from La Barceloneta, then Barcelona, then Catalonia, and this is very clear for us. Okay. The second thing is that we are an institute that we work on environmental sciences, in particular to the sea, but environmental sciences, so that we have this social commitment that is very clear for us. So we are working in ocean science, but as you can see when you are in our hall and we have this folder, we work also on ocean science for a healthy planet. And this is a little bit our lemma, our motto in this institute. Second thing to know is that we are the largest marine research center of the Spanish Research Council, I would say of Spain, because I don't think that there is other center of our size, except maybe the Instituto Español de Oceanografía, but this is not a single center. They are distributed all along the country, so there is not a single center as large as this one in the country, okay? You will see the size in a minute, but what is very clear for us, and I think it's important for this meeting, that we are very multidisciplinary. Here there are groups, there are departments working in, I would say, most fields of marine sciences. So here there are many biologists, because our history comes from mostly from biology and fisheries, but there are also physicists, there are chemists, there are also not mathematicians. I'm not sure if there are mathematicians here. Are there? Used to be one, but not that much. And this is why it's very important to have this meeting, you know, because we have luck here. When we concentrate in scientific output, we are clearly, we have a leading role in Southern Europe and in the Mediterranean region in terms of scientific output, projects, papers, and all this. But when we compare to the Northern Europe, we are comparable also when we normalize for the number of researchers that we have here. There are larger centers in Northern Europe, but when we normalize, we have more or less the same output. And then, thanks to this output, we got the Severo Ochoa accreditation 3 years ago, so we have to renew it next year. We are working hard now to have a good project for the next year, a good strategic plan, okay? I was talking about social commitment. I will repeat this a few times during this presentation. We are a center with a very strong social commitment, so we think of excellence as a multidimensional concept, in which one is, of course, having ERCs or having very publications in high impact journals or whatever, but also for us, excellence is getting our research having an impact on society, be able to talk with the administrations so that the decisions that they make are based on scientific evidence that we can provide, and we work a lot with them. And also to have an internal organization so that people here is happy or wants to come here, and all these things for us are components of excellence, okay? And this was explained more or less in a book that we can give you, or give the PDF that we published a couple of years ago, in which we explain a little bit the kind of science we do and the kind of objectives that we have at all levels, purely scientific but also social. We are, as I told before, we are centered in the Spanish Research Council. The Spanish Research Council, even if you are not from the CSIC, you know that it's the largest research organization in the country with over 15,000 people in the country. And we are, I'm not sure, but I think we are the fourth or fifth larger institute, so we are one of the largest in the CSIC, because the CSIC has over 120 institutes. It changes from day to day, it depends on who explains it, says it's 120, 125 or something. Mostly our own, as we are a propio, un centro propio of those 68, but there are many also that are mixed with universities, with comunidades autónomas, with other things, okay? From those, there are 11 Severo Ochoa and 5 Maria de Maeztu. We are one of those Severo Ochoa. We are an old center with a long history that comes from the 50s. In 51, there was this, at that moment, it was called the Instituto de Investigaciones Pesqueras, that had an aquarium, which was very well known in the city. Here in La Barceloneta, it was visitable, and many people had been there before. So we were at this building that you can see at the left until 2001, okay? But it was, and we have a number of highlands, different type of highlands. For example, it was people from this institute who started with the Antarctic base at that time, and for example, Pepita Castellví is very well known. And scientifically, there are very well known researchers in the history of ICM, and for example, above all, for example, the person that gives the name to this, this auditorium where we are, it's Ramon Margalef, one of the very best well-known scientists in Catalonia of EBA. Then we moved here in 2001, and there has been a number of things, and of course there is many people working on ecology, on fisheries, on different aspects, but there is also people that have led missions from the European Space Agency, for example. And we got the Severo Ochoa 3 years ago, as I was mentioning. Then we have been, one thing about the Severo Ochoa, maybe it started a little bit before, but of Severo Ochoa, is that we have been increasing a lot the number of people that we have, especially with postdoc, Ramon y Cajal, and things like this. But now, this year, at the end of 2022, we were 314 people, and 4 years before, we were about 250. It had been stabilized at this number for 10 years, and now we are rising again in the last 4 or 5 years, the number of people. And you can see that there are more women than men, but we have a number of structural problems. For example, we have this scissor effect, and very clear, in which we have more women than men in some positions, but when you go to the upper positions of professorships and this, there are much more men than women. And we have a number of measures, a gender equality plan, and working on this, but we have still a long way to go. I won't explain this because it's a bit complex, but I must say that we have quite a participatory structure in our governance. We have a number of committees, a number of task forces, a number that we have a lot of meetings, which made our governance quite complex and requires a very rich ecosystem in which that requires a lot of energy. But still, we think that it has provoked a change in the way the people perceive the institute, and we think this is that has more positive than negative things. And for example, these are a number, 3 examples of task forces that work a lot in the institute that are very propositive and that made a change in the institute. For example, the Quality Task Force, it has worked a lot the last years. We have a gender equality plan, which is an example for CSIC. They take it as an example many times. And we have had several years ago this Distintivo de Igualdad for CSIC, the Sustainability Task Force, and one which is probably the first one that was working, it was the Marine Science Literacy Task Force. We have a lot of effort on this. So going to the society, education, these kind of things, okay? You will see a little bit more in 5 minutes. Just to have an idea of our size regarding distribution regarding funding. Last year we got almost 20 million from which more than 60% is money that we got from projects, and 39% last year it was the basal funding that we have from the salaries of the people, which is civil servant, and some money that we have to run the institute. But the rest comes from projects that we got. And you can see that last year it was more or less equilibrated between national and international projects, mostly Horizon Europe, and then a little bit of knowledge transfer. But this knowledge transfer, the last year, it has been increasing a lot because we are having big projects, especially with the Generalitat. I will explain a little bit in a minute. I won't go to the detail of our publications, but just one example for you to know. When we concentrate in the Nature Index, which is this selection of that made the Nature Publishing Group of the journals that they consider most prestigious for them, it's 82 journals. And if we have a look, we have always the largest nature index in the Mediterranean and Southern Europe and in Spain, okay, of all the marine and environmental centers in this area, so that you know. But again, now, and especially in the new Severo Ochoa, we are concentrating on changing a little bit this idea that we have of what is science of excellence, that before we had a kind of dichotomy saying we are making science of excellence, but at the same time we have this social commitment. We have changed it now, and now what we think and what we are trying to put very clear in the new Severo Ochoa is that for us the excellence should have integrated the social commitment. So there is no excellence without social commitment, without in a center as ours. What do we understand by social commitment? Is having this marine research in which we have also knowledge transfer, and we work with ocean literacy, and also we try to strengthen our ties to the territory. And for this we have 3 programs that we run, and I won't present it in detail, just a little bit. 3 programs. One is has been running for a long time, the other 2 are quite new, which are ICM transfer, to foster the knowledge transfer to the private sector and to the public administration. ICM Divulga, this has been running for 10 years, more than 10 years now, almost 20 I would say, is promoting outreach, education, citizen science, and now lately art and science also. And for this is very important for us, I repeat. And another one, which is ICM Cares, which is the internal part, improving work environment and talent development consistent with our values, okay? Just a little bit about how we organize ICM transfer. We have made a portfolio that we have, which is structured in 4 thematic areas that you can see here. It's sustainability of renewable living resources, the observation and the modeling of ocean and climate, the health of marine ecosystems in a wider perspective, and then the protection and dynamics of the coastal environment. This is our portfolio of the things that we know that we can transfer to the society, to the administration, to the enterprises. On each of these axes, we have collaborations with enterprises, with administrations, and something. For example, the most important one now in terms of size is an agreement that we have with the Generalitat de Catalunya to create a new structure, which is called ICATMAR, Institut Català de la Recerca per la Governança del Mar, which is this agreement that we have with the Department of Fisheries of the Generalitat, that they are giving us funds for 6 years now to develop these 2 first lines. So we are working with them in the governance of the marine, of our coastal system in terms of the sustainability of the living resources. There are scientists and technicians from here that go with the fishermen to the sea, and they provide data to the Generalitat so they can manage the fisheries. And the second, and something that was lacking before, is the system of observation of the ocean data. For example, a number of radars for the currents, and a number of instruments to measure temperature and other things that are controlled from here, to have predictive models on the state of the sea, that come together with the fisheries modeling. okay? The other two, there are more initiative of single researcher or groups with enterprises or something, but we would like to extend this relationship that we have with the Generalitat to the first two, to the other two, because we know that we can provide a lot of information on this, okay? And the other thing, just a few words, because we are quite proud of this, is ICM Divulga, is promoting as outreach, education, citizen science, art and science. It has these 4 things, and this has been developed for a long time. It started in the early 2000s, more than 20 years ago, with the first online campaign diary. Then ICM Divulga was created, and we have had a lot of projects on. And now, for example, there are researchers from here, one researcher from here that is coordinating 3 projects of citizen science, and then have a lot of projects of education. For example, we have these petite oceanographs, in which there is a meeting here every year in which you have hundreds, this is full of kids that come here and present their works and all this, and this is very important for us, okay? And it's as important for us that we have been talking, and this is one of our main projects, thing that we want that will happen, and we hope that will happen, is to have a space fully dedicated to marine knowledge transfer, education, and outreach. So maybe you heard from the newspaper something that we are trying to get Opium, which is a discotheque that is just here, and this is going up and down, but we are quite confident that it will happen next year or something, and to have 2,500 square meters in which we would have all these activities, all the projects that are to the society in the same space, okay? And this will be visitable, will be something like this. One space with knowledge transfer to institution and private sector, in which we would have, for example, the headquarters of this ICATMAR and other. The second for marine education, in which we have projects and working with the Centro de la Cultura Contemporánea de Barcelona, the Thyssen-Bornemisza Museum, and others. We are talking on how to do this. Then we have the projects of Ciència Ciutadana, of citizen science in another space, and the last one would be community participation. The projects that will be there, we know what they are. In the next 3 months, we should have the project, because now we will know in a few weeks which is the company that will do all these works. Okay. And I will finish with the thing that is more important for this meeting, just explaining what our research lines, and just a few words of the projects that will be presented the next days. So our organization of research is in 3 main research challenges. We are now rediscussing this for the next Severo, but as it was in the other one, it was these 3 research lines. One concerned to life, in the sense is the conservation and the sustainable use of marine life and ecosystems. The second to climate is the understanding of the role of the ocean in the climate and the interactions between both. And the third is on hazards, is the comprehension and mitigation of anthropogenic and natural hazards. This is the stable organization of the research lines. Then there are research lines which are halfway between some of these things. As you can see, there are different research lines, but this is moving a little bit. We are re-evaluating every time which are the research lines that go for this. And you can see that the way we are organized with respect to these challenges is here. We have 14 research groups that belong to 4 departments, but those departments are mostly administrative. And the research is not done by the departments itself, but by the groups that you can see here. The departments are the ones that were several years ago: marine biology, Department of Physical Oceanography, marine biology, renewable resources, and geosciences. These are the groups, but you can see that most of the groups, as you can see here, they are halfway between different, they contribute in different way to the different challenges. You can see that the ones which are more in the center, they consider that they contribute in their research lines to the 3, and the others that are at the limits, they are more focused on several of those. And here, just a little bit, the things on climate, there are the lines that we work on climate is to understand ocean dynamics and its role on the Earth past, present, and future climate, and understand how climate-driven processes and global change affect the state of the ocean, including marine life. And on this, there will be two contributions on this, or mostly one. One is that on the use, most of them are concentrated, they try to use machine learning or artificial intelligence. So this one, for example, that will be presented, I don't know if it's now or it is today or tomorrow, the next day. This will be machine learning for wind surface forecast, okay? There will be another one to try to use artificial intelligence applied to the tracking of marine vehicles also. In the second thing that we will see in the part of life, we have also three lines related to this, and we will have three different contributions, one that has different aspects related to epigenetic changes in fish, one related to the development of epigenetic clocks, another on epigenetic biomarkers, and another on epigenetic inheritance to improve aquatic production. The second one is they are trying to predict microbial functional redundancy, and the last one on life would be trying to predict animal movements, to taking into account the resources and in response to the environmental forcings that we have, okay? And the last one on hazards, there will be also two contributions, one more related to mimicking tectonic processes by mathematics, okay? This is related to the implementation of mathematical expressions on numerical formulations to understand tectonics. And the second one is deep learning applied to different things. One is, for example, a classical problem in which we have a signal that we want to identify in a very complex and noisy recordings of seismograms, for example. And the other one extract more information on the models that we have, also using machine learning. So that's all, and I will just show a bit our institutional video so that you will know a little bit how we see ourselves.
What do you think about when you think of the sea? Maybe about how peaceful it is, or perhaps about something that fascinates you, that really stirs your imagination. Or perhaps you just think about a vast stretch of blue with an eternal breeze, or a smell. Or maybe it conjures up all kinds of memories. For some, it's an idea, a great mystery. But for us, it's a great story to tell. The story of how life finds a way, even thousands of meters deep. A story about tides, underwater currents, and unexplored depths. A story of how, on our planet, everything's connected. A story about us and what we're able to do together. And what we're still not able to do. A story whose ending has yet to be written, told from the shores of the Mediterranean, full of magic, and every day, a bit more exciting. What do you think about when you think of the sea?
Gracias and welcome again.
Thank you very much. Valentí forgot to say that he's the director of the center, but I guess you realized. Welcome, everybody. It's a great pleasure to have you here today. We have been working hard to make this happen, and I would like to thank Luis and Josep and David and also Sonia that have been organizing all these events. So today we can be here. We are going to have 4 days. That's our aim is that we get to know each other, we get to know what we do, and you will see that from the ICM perspective, hopefully we convince you that we do very interesting research, lots of data, lots of questions, lots of challenges, and sometimes we are actually challenged by the methods and the mathematics that we have, the mathematical tools that we have in hand. So we are willing to, that you fall in love with some of our work and some of our challenges, and hopefully we create some alliances, some projects together, maybe some students collaboration. So please come with your minds open and discuss with all of us these days. I think ECM researchers, I think I can speak in the name of everybody, are a bit nervous because we are not mathematicians. And some of you told me, oh, but I don't know what to speak, I don't know what to say, all my talk will be wrong. I say, well, just relax, okay? And from your side, mathematicians that are coming to see us, also have a little bit of patience with us. Sometimes we explain things maybe not the way we are supposed to do it, but these 2 days are exactly for that, so that you can help us, we can also help you to pose you new challenges and show you all the data and all the important questions that we are aiming to answer from the Institute. So that being said, we have 4 days, 2 days here, so today and tomorrow, and next week, Thursday and Friday, will be at UAB in the Center of Research for Mathematics. And we have decided, so when we are discussing the program, we have decided to have a lot of room for discussion. So we are going to have 2 or 3 talks per day in each session. So 1 session, and then we'll break for discussions, and then coffee break, and then we'll have a lot of opportunities to interact. There's also some posters that we have from students and researchers that are today and tomorrow will be in the main place, where we will have also the coffee break. So take a look and interact with the people that will be hopefully in front of their posters and around. Okay, so we are going to start the program. Let me know, today I will moderate a little bit the session with help from Gonzalo Cimarro, and tomorrow Gonzalo will take the lead and I will be helping around. And then in CRM we'll have moderators. David told me that and then you will moderate the session. Okay, we are going to start then our session with unlocking complexities in the ocean microbiome and fish genomics using math. I think it's Francesc Piferrer and Pierre Ramond that are going to speak. I don't know if Pierre is going to be first. Yeah? Okay, thank you very much, and welcome once again to ICM.
Well, hello everyone. I'm Pierre Ramond. I'm a Marie Curie postdoc fellow here at the ICM, and I work with Ramiro Logares. First things first, thank you for inviting me to do this presentation. To be fair, I'm replacing Ramiro, so don't judge if it's not too much on the math side. Today, I'll talk about my research field, which is microbial functional redundancy and microbial functional ecology. And I'll talk how we use math to within this field and to compute and predict the future of microbial functional redundancy in the ocean. So in this talk, I'll talk about microbes. Here I'll focus mostly on bacteria and archaea. Here you have a tree, a phylogenetic tree, which represents the various branches of this life domain. So you can see that there are many branches because microbes arose like 4 billion years ago and diversified since then. So you can see many branches, but also many and various forms on that tree all around it. And one thing microbes are also very well known for is their metabolic diversity. So if you think about earth elements, so what we call biogeochemical cycles, most of those biogeochemical cycles, they are driven by microbiomes and microbes. So an example, carbon, sulfur, or nitrogen, they change from one form to another through the microbiome compartment. So in that sense, microbiomes, they have a super important role in ecosystems. One thing that we discovered recently is that also microbes are functionally redundant. What do we mean by this? Well, if you look at this picture, on the x-axis, you have various functions, so metabolic pathways that are involved in biogeochemical cycles. And on the y-axis, you have microbial taxa, so a lot of diversity of microbial taxa. And if you have a look per columns, okay, you can see that many different taxa are able to perform the same function. So this is what we call functional redundancy or metabolic redundancy. Now, there's some hypotheses about the role of this functional redundancy in the functioning of ecosystem. The first hypothesis is the one of insurance hypothesis. If you were to kill one member of the microbiome, basically, because of this functional redundancy, one other member of the microbiome could replace it and perform the same function. A second hypothesis is really related to the first one because it talks about the stability in time of the functions performed by the microbiomes. Basically, by having many organisms being able to perform the same thing, the function can tolerate disturbances in the environment and maintain a high flux for the biogeochemical cycle. Okay, so we know this, but what we don't know is patterns of microbial functional redundancy. We don't know where it's higher, where it's lower. And this is quite problematic because we saw that microbiomes have a huge importance all over the world, and especially in the ocean. But we know also that the world is going to change as we know it because of climate change. But we don't know what will be the effect of climate change on microbiomes and this functional redundancy. So in this talk, I'll talk about how math can help us explore this topic. I'll first talk about metagenomics bioinformatics and how they allow us to build the field of microbial functional ecology. Then I'll talk about how we can quantify microbial functional redundancy in the global ocean. And later on, I'll talk about machine learning and how I've used it to predict patterns in the future ocean. So, metagenomics bioinformatics, strong words. I'll try to go easy with this figure. Basically, here at the ICM, what we would do is that we would take a boat, we would cross the ocean, we would sample water with those knee skin bottles that you can see here. This water, we would filter it on filters that target the microbial compartment. And then we would freeze those filters to maintain zero microbial activity and maintain the microbiome as it is when it has been sampled. And then once we go back to the lab with various chemical lysis and everything, we can break those microbial cells and extract their DNA and isolate it. And later on, what we can do is sequence it, okay, with next generation sequencing with various methods involving short reads or long reads. Okay, so now we have the DNA of our microbiomes. The first thing that we can do is to try to reassemble the genomes of populations of microbes, okay? So we have populations from the same species, and we try to reassemble all the genome of those microbes. So just as a recap, we sequenced our DNA. We now have various samples and various reads. So reads are short fragments of nucleotidic DNA, okay? So to assemble those reads, we do what we call co-assembly, and this relies heavily on math. And most notably on what is called De Bruijn graphs. So to explain shortly, and I'm not an expert in this, De Bruijn graphs are a sort of method that applies a sliding window across the nucleotide. Here we have a window of 4 nucleotides. So we are dividing this read here in a windows of 4 nucleotides that we are sliding across the read, okay? So these are the 4, that's what we call k-mers. In that case, it's a former, okay? And we do this for each read. And based on shared k-mers, so as you can see here, those 2 reads in bold, you can see that they share nucleotide k-mers. So that helps us to reassemble the reads like 2 puzzle pieces, okay? Of course, sometimes it's not only 2 reads, but it's a higher number of reads. In the situation below, we have 5 reads, but we also have variability, which means that, yes, we can assemble those reads, but sometimes there is inner variability in the nucleotidic sequences. This inner variability, it represents the variability in the genomes of the various members of this population of the species. Yeah, okay. I'm not going to detail too much this slide, but it's the first step that I presented. Is one of the many steps that allows us to recreate microbial and population genomes. Okay, the end product is nearly complete microbial genomes with low contaminations, which means that genes originate mostly from the same population. And with those metagenomics assembled genomes, or MAGs, I call them genomes. You have also taxonomic information and phylogenetic information, but also metabolic information. Another thing that we are interested in is, okay, we have reconstructed all those genomes, but we would like to know where they are found, okay? So to study genome distribution, what we can do is competitive mapping. So like I said, I'm not an expert in reconstructing genomes, but what I did was that I downloaded a database of microbial genomes from Luca Paoli and co-authors, so that's the left side of the graph. And I did what we call mapping, which is trying to check whether reads from metagenome sample from all over the ocean, if they map onto the genomes that we have in the database. So this is kind of a complex bioinformatics section, but basically what this gave us is the number of reads per kilobase pair of genomes per GK-base pair of metagenomes. Well, if you need to take a message from this slide, it's just that by doing this competitive mapping, we have a genome distribution across all those samples in the ocean. Okay, so we have genomes, we have their abundance in the ocean. Now, what we are interested in is what is the functional potential of those genomes. And this we can do it with gene and functional prediction. Here I've used a pipeline called metabolic that focuses on genes that are related to enzymes or proteins that are involved into biogeochemical cycles. It's a complex bioinformatics pipeline, but basically it relies on an existing database of genes and proteins for which we have a sequence, and we know the role in the metabolic role, or the role in biogeochemical cycles. So trying to predict those genes and to map them to the database, we use what we call profile hidden Markov models. So once again, I'm not an expert on this, but this is a very useful tool for us in our field that you probably know more of than I do. So this allows us to know, okay, what is the role of a microbe in the ocean. And this allows us also to create a trait versus species table, okay? So we have a description, a functional description of our genomes. Okay, so now that we have on one side the genomes and microbes distribution across the ocean, and on the other side a trait description of the functions of those organisms, what we want to do now is to study their functional relevancy across every sample. So to do this, I reviewed the literature, and that was a very interesting review from Carlo Ricotta and Sandrine Palvoine and their co-authors that studied ways and methods to study functional relevancy. So first things first, here we're going to talk about traits. And traits are any measurable feature that is a proxy of an organism's role in the ecosystem. So if you remember the last slides, basically we're going to use those genes involved in biogeochemical cycles as traits. Another assumption of those methods is that species with similar traits are assumed to support the same function in the ecosystem. If they share the same genes, they supposedly have the same role in the environment. So within this review, first the authors reviewed existing methods based on trait-based distance. So if you have a look at, can you see this? Yeah. If you have a look at this plot, basically species can be put into a two-dimension space, and they can be placed into that space based on values they have on their traits, okay? And two species are close if they have the same values in the trait, or far apart if they are very functionally distant. This functional space, it can contain various traits, many traits, and it can also be divided here based on different cluster size, and within those clusters, basically species would have the same function. But this is not an actual metric to measure functional relevancy. We could check the richness within those clusters, but it's not a unified metric. So what these authors did was suggesting a method based on trait-based distance, where species distance between them equals 0 when all species are equal, and equals 1 when species are distinct. Based on this trait-based distance, what we can do is compute hierarchical clustering dendrograms. And the method relies on the relation between species on those trees. Basically, if you have a related tree, that means that you have species that are very close functionally. On the other side, if the branches of the tree are all independent, that means that all species are distinct. And based on the shape of this tree, they developed a metric that equals 0 when all species within a sample are functionally distinct, and equals 1 when all the species in a sample are functionally equal. Okay, so now great, we can actually compute functional relevancy in the global ocean with this method and our previous data. Okay. So these are the patterns of functional relevancy in the surface ocean. As you can see here in dark blue, what we observed was that functional relevancy in the surface ocean was higher in the equator and the subtropical parts, and lower in the poles and the temperate ocean. If we have a look at the correlation with environmental variables, we can see that the main gradient is a differentiation between conditions in the poles and conditions in the equator. So lower temperature in the poles, higher nutrient concentration, or light limitations. Okay, but this analysis is good, but we can do better. We can do better with what we call machine learning, and here I'll talk mostly about neural networks. if we have a great correlation between functional relevancy and the environment, we can try to build models that predict functional relevancy based on the distribution of environmental variables. So I did this with a R package called NNet. And basically the pipeline is that I took my dataset, I divided it in two. I trained a neural network to study the relationship between functional relevancy and the environment. And then I could cross-validate the quality of the prediction of those neural network models based on the second subset, so the validation dataset. I repeated this 1,000 times, and I got 27 good neural network models that predict functional relevancy based on environmental conditions. Okay, so we have good models to predict functional relevancy, but we need to know the distribution of environmental variables in the ocean. So what I did is that I downloaded data from the coupled model intercomparison project. What is this project? It's the project that compiles all the Earth models data that the IPCC relies on to make predictions for the future climate conditions. Okay. So I downloaded data for those 9 environmental variables in 2015 and in 2100, and according to different climate change scenarios. So basically, the baseline in 2015 is equal across those scenarios, but in 2100 they differ because it's different scenarios of climate change. So now that we have values of those 9 environmental variables, we can try to predict functional relevancy in the surface ocean. So those are the results of the 2015 estimation. So we see and repeat sort of the results that we evidence on the first map, where functional relevancy is lower in the equator and the tropics. No, it's higher in the equator and the tropics and lower in the polar and temperate zones. So if we come back to our fundamentals, supposedly temperate and polar ecosystems would be less insured against perturbations, disturbances, or extinctions. Now, let's have a look at what's the future of functional relevancy. So here we have 4 plots. They are all divided based on the different climate change scenarios. Here on top of the plot is an estimation of how many degrees we're going to have compared to the industrial era. So you can see that this is the less harsh climate change scenario, and this one with the plus 4.4 degrees is the harshest. The first observation that we have is that functional redundancy, here you have a delta between 2015 and 2100. So we can see the variability in the future. So we see a small increase of functional redundancy in the southern pole, so with that blue layer here at the bottom. But most importantly, we see a decrease of functional redundancy in temperate zones and the equatorial Pacific. So if you have a look at the red layers, it's exacerbated in the harshest scenario, but you can see that all the North Atlantic or the boundary with the Southern Ocean all go into the red part, so we're going to lose microbial functional redundancy. So if I were to have some conclusions for this talk, metagenomics, bioinformatics, and multivariate analysis allowed us to quantify microbial functional redundancy across the ocean. At surface, the temperate and polar zones of the ocean showed the lowest redundancy, and the climate change projections suggest that further losses of functional redundancy in temperate regions by 2100. This is quite important because temperate regions, as you can see on this map of the fishing effort in 2022, or I forgot, but basically temperate regions, especially the North Atlantic, are heavily fished, and so if we have a change of ecosystems and the microbial ecosystem, perhaps we could expect those ecosystems to be highly impacted by microbial change. And the question that I have for you, and that will probably be my conclusion, is that could this ecosystem shift towards an unwanted state, and that we wouldn't be able to come back to in the future. So thank you, and if you have any questions, I guess we can have them later.
Shall we do some questions now, or yeah, maybe not? Any questions?
Hi, I'm interested in how you, in the way that you reconstruct the full genome given the reads.
Mm-hmm.
Do you have some software or some algorithm to do it, or I don't know what's the process? So
it's a lot of softwares that have been published. I don't develop this kind of thing.
Okay,
like
so there are a lot of existing.
There are a lot of tools,
okay.
And actually there are tools to combine all the tools, because all the tools have their own specificities. So sometimes what we want is like a mixture of all the quality outputs from those aligners.
Okay.
Mm-hmm.
Yeah, I have a small question. It's about the model you have been studying with this neural network.
Yeah.
Is it difficult for this model to make mechanical or a deterministic for this problem? I mean, to make a deterministic model to study it? By deterministic means something of dynamical systems nature, that there is a deterministic evolution in time instead of using neural networks that in some sense we do not know what they are doing.
Yeah, actually, so what we do with those neural networks is just predict the future niche of functional redundancy. Basically, where we saw it's higher at the moment, we project those conditions in the future, and basically functional redundancy is just gonna, what the neural network does is just like moving or lowering redundancy based on the future conditions. But what this doesn't take into account is obviously the adaptations and the evolution of microbes. So for this, what would be very interesting for me would be to work with people that do work on Darwin models, which are models of evolutions of microbes and plankton in the ocean, to compare our results with their results to see how evolution is going to affect the patterns we observe.
Okay.
Very good question.
Nice idea.
Yeah.
More questions?
Thank you. Do you know, or are there some research relating this functional redundancy, if it correlates with higher diversity of, let's say, higher level trophic levels? Is there any relation? Because at least, so you have like different species that are doing the same function, so it perhaps has an impact on biodiversity at the other trophic layers.
Yeah, definitely there are some papers related to this, but when we focus on the microbial compartment, we mostly focus on functions that we can measure, which are easily measured. Well, not so easily, but we try to check the biogeochemical fluxes. So I think most of the research tries to correlate functional redundancy to this and less to the higher trophic levels, but that would be definitely a great thing to explore. I think there are some papers, but I can't tell.
Any additional question? Okay. Well, we are changing the presentation. I have a last question. You were using those TMIP6,
Yeah.
I guess this is the ensemble of the models, or did you choose? I missed that if you choose a specific model or you use the ensemble.
No, I merged the output of models that had the same grid.
That have the same?
Grid?
Okay.
Yeah.
Okay.
So I merged like the net CDFs.
Okay. Maybe it would be interesting to try to see which ones, because some of them are quite different.
Yeah.
So maybe the extremes, you could also try to run those with the extremes and see how that would change.
I try to have like a clear message, but you can obviously explore all the models separately.
We do similar things with higher trophic levels, as you were, Josep, mentioning. So let's discuss even within ICM.
Sure.
Nice, thank you. Okay, so we are going to change the presentation. Francesc, you want to? The schedule is we have an hour for the next presentation and discussion, and then we have a coffee break at 10:45. Okay. So just wait a little bit more for the coffee.
Good morning and hello. Thanks for the opportunity to talk at this workshop. So I'm going to talk about the challenges in assessing the genome-wide, developmentally dependent and environmentally induced epigenetic changes in fish. And this is the work that we have been carrying in our group in the last years, and especially is the work of the past and present members of the group, Daphne, Alejandro, Nuria, Silvia, and Omar. And we have three specific problems that I want to share with you. The first problem is the difficulty in knowing the uncertainty in outcome variables, an amount of non-informative predictor variables when we deal with problems in where we have like the typical situation where you have that the number of variables is much, much larger than the number of samples that you have. And this is important in the context of the development of epigenetic clocks for fisheries management and biodiversity conservation. This is one type of problems. Second type of problems is the dealing with low sample sizes and for machine learning-based methods applications. And this has to do with the use of epigenetic biomarkers, I'm going to show you what it is, and their integration in breeding programs in aquaculture. And the third typical problem is the proper estimation of the amount of epigenetic inheritance. And this is something that not only we have problems in terms of like methods to use, but also in the concept itself. This is a very hot topic in biology, in the debate of nature versus nurture, and on the context of what is called the extended evolutionary synthesis. And here we're dealing with the epigenetic inheritance to improve the aquatic production and also to assess how organisms may cope with a changing environment. So I have to say that from left to right, so problem one, it's a difficult problem, but we think that we are in the way of how to know how to solve it. Problem two is a problem that we have proposed a way to solve it. And problem three, again, is something that not only is an issue of the methods to use, but also of the concept itself. So the general context of this started, believe it or not, with the Human Genome Project in 2001. And this was an era of optimism. No? It was like the whole genome of the humans, and therefore opened the road for other species. It was like sequence, and candidate gene studies could be done at unprecedented resolution, at single nucleotide resolution. So you could theoretically know what would be the contribution of single nucleotide polymorphism in a character. Traits could be mapped to specific genes and genetic variants. And this, again, was a very optimistic situation. But then, sooner after that, there was the problem of the missing heritability, which started to become evident in about 2008. And this is about the fact that these single genetic variations, like for example the SNPs, could not account for much of the heritability. And sometimes this heritability was assessed to be at 0.8, which is huge, which is very high. And this was particularly relevant for the situation of complex and continuous traits. For example, like diseases, behaviors, and other phenotypes. So when does this missing heritability comes from? In 2012, there was the ENCODE project, the encyclopedia of DNA elements, where all the regulatory elements in DNA was put forward. but still was not the solution for that. Now we know a lot for how the genes are regulated, but still is not the solution. So there are different proposals to to see where the missing heritability comes from. One of them is like, for example, unidentified sequence variation. For example, copy number variation, large genomic rearrangements, rare mutations, or rare alleles of large effect, for example. Then also genetic interactions with epistasis, epigenetic variation, and this is what I'm going to mention now, and so forth. No? So, and now we know that a lot of complex traits are very polygenic. And this means that, for example, for these complex traits like height or weight or IQ or schizophrenia, things like that, the results now, what is evident, is that only can be assessed, the different SNPs, the contribution of different SNPs can be only well assessed with sample size that sometimes are more than 100,000 or 300,000 samples. And this is possible in biomedical research because they have access to many samples, but this in ecology or evolution is completely different. So this is one of the problems, the availability of sample sizes. So epigenetics, as one of the possible solutions, is the study of mechanisms and phenomena that are related to heritable changes in gene expression that are not dependent on DNA sequence, so they're not mutations. So we have three epigenetic mechanisms: DNA methylation, the addition of a methyl group in a CG context, only in a CG context. Basically, there is other possibilities, but more than 95% is this in vertebrates. Then histone modifications and the RNA interference. I'm going to talk about DNA methylation most of the time. So this is a code superimposed on the genetic code, which means that this is a sequence that is not methylated, that is an enzyme, DNA methyltransferase 3, that methylates, and this is the de novo methylation. So to write this code on top of the genetic code. And then there is enzymes that maintain this methylation when the cell divides. So these methylated sequences can be inherited through cell division through mitosis. And then the same way that you can write and copy this information, you can erase the information. And this can be active demethylation through the concourse of 10-11 translocase enzymes, or simply passive demethylation, simply without the action of one of the enzymes that maintains the methylation. So basically, in the genomes of all organisms, this is what's called epialleles. These are identical alleles that have different epigenetic modifications. So for example, here you have the promoter region of a gene, you have a CG, different CGs, and these CGs cannot be or can be methylated. When this in a different in a specific loci you have different methylation, then this CG is referring, you'll see it as a CPG, this is the notation, cytosine phosphate bond 1-in, and then this can be methylated or not. Then when there is a statistical difference between the level of methylation here and here, between two conditions, for example, you speak about differentially methylated Cs. When there are many together, you speak about differentially methylated regions. So you'll see along the talk these two abbreviations. And this regulates gene expression. So the typical dogma is that when this is not methylated, the genes are on, and when this is methylated, the genes are off. But there are many exceptions to the rule. So this level of methylation depends on both internal and external factors. And one of the most important ones is tissue. So the genome is the same in all of the cells, but the epigenome is different in each one of the different types. And we have in humans, for example, more than 200 cell types. So you have like same epigenome in the blastula, but then you start doing organ differentiation. This is made possible because the same genome acquires different epigenetic programming or different epigenetic patterns. And this can be changed according to some environmental conditions. So epigenetics is very important because it's a bridge between genome and environmental information. It captures all the environmental information, and for the epigenome, the environmental information means not only the environmental outside the organism, but also the internal environment. Okay? So we have different factors that, again, internal and external, that contribute to the shape of the epigenome. So again, and this is very important, we have, each organism have one typical DNA sequence, each one of us is different, but each one of our cells have a different epigenome. So this is a very challenging situation because when you address a specific problem, you have to first to choose what tissue you will use for your analysis. This is very important. If you, for example, try to assess the importance of, for example, alcohol consumption in your body, and in terms of epigenetic modifications, you would look, for example, at the liver, not at the brain, this type of thing. So, and another thing that is very important is that this epigenetic information, like a computer, when you reset the computer, this is all erased. Typically, this is all erased. This is at least the dogma in mammals, no? in mouse. But as we are seeing now, in fish, it seems to be like quite a lot of difference. You know? Some fishes have little reprogramming, and some fishes have essentially the same thing as mammals. So that means that some of the epigenetic marks that can be induced by the environment in the parents can escape this reprogramming and be passed to the next generation. So therefore, there is also inheritance through gametes, through meiosis. And the important thing about aquatic animals is that in contrast to mammals or to birds, for example, where fertilization is internal and early development is internal, in mammals or in birds, internal, external, in aquatic organisms, no matter what, these ones are the importance for production, for aquatic production, like mollusks, shrimps, and fish, everything is external. So therefore, the sensitive stages of development, which are during very early development, in these animals, you can manipulate the external environment to your convenience to induce epigenetic changes that can be related to a phenotype that can be of your interest, for example. We'll see that. And one last thing I have to say is that of these different types of epialleles, there are some of them that are totally linked to the genotype, so they are not informative. They don't bring additional information to the genotype. These is called obligatory epialleles. Then there are some of them that at different environments, and depending on the genotype, they have different behaviors. And there are some of them that are pure. That means that when there is a change in the environment, and regardless of the genotype, the response is the same. So what we think is that what we want to do is to identify epialleles that are pure, like these ones, that heritable, can be passed from one generation to the next, and also that are linked to the phenotype. Okay? And this leads to the development of epigenetic markers. This nowadays is very common in biomedicine. There are commercial kits already based on DNA methylation. And you take a DNA sample, it's a PCA-based method, and they have a high efficiency. Okay, so if you look at these things, like 10 years ago, you would see that these kits are based on DNA changes in sequence, but now they are based on DNA methylation. Why? Because DNA methylation has one extraordinary property, which is that they have only not diagnostic, but also prognostic value. So that means that you can predict what's going to happen, the outcome or the susceptibility of that person to a type of cancer or whatever. And that's what we want to do, to see how animals will face different environmental situations, in the face, for example, of climate change. So epigenetic markers not only have the diagnostic but prognostic. So here you have a situation in where you have a control, and then you have an external input like this red arrow, and immediately this is captured by the epigenetic landscape, but not captured by transcriptomics. So you have like the situation in which at time one you have changes in the epigenetic landscape, but not in the transcriptome, and then situation in which at time two both are present. So this one is prognostic of what's going to happen later, okay? And then, so you have this combination of genetics and environment. You have, like, for example, this gene. You have different CPGs. This is due to temperature. You have the level of methylation goes from 0 to 100 in low temperature and high temperature. And combining these differences, then you can develop epigenetic marker. Okay? So you have genetics and environment, the epigenetic marker, and this is translated into changes in phenotype. For example, here in change of expression. So again, this is being developed for biomedicine, but our question is, what about the use in food production, agriculture, livestock, in our case in aquaculture, and what about their use in natural resource management and biodiversity conservation? And for this, usually you have you start with genome screening for candidate loci or features, and you use techniques like whole genome sequencing. And then as soon as you progress with the development of the markers, you go with the genome-wide, but not whole genome. And finally, you go with targeted approaches, locus-specific. And then the epigenetic modifications that you target, that you'll see in a moment, are many. And then you filter until you get the ones that are most informative. And at the beginning, you have to deal with, this is the application, but here although it seems that there's few sample sizes, that's a problem that was mentioned before, that sometimes to capture all the variability, you need to start with many, many different samples. And when you apply machine learning procedures, there is supervised and unsupervised methods. Basically, the ones that we have used is these ones, classification and regression programs. Classification for characteristics like binary characteristics like male, female, or health or disease, and regression line for growth and age, for example. And these are some of the epigenetic markers that have been developed in the CBAST in the last years, okay? So the first problem is how to deal with knowledge or uncertainty in the outcome variable and the large amount of non-informative predictors. And this is like finding a needle in a haystack and has to do with the development of epigenetic clocks for fisheries management and biodiversity conservation. And what are these clocks? So in all vertebrate genomes, during aging, there is a slow but consistent decrease in the level of global methylation, all right? But against this background of global genome demethylation, there are some loci in the genome of among the millions of loci in the genome. For example, the CBAS has more than 10 million CG loci in the genome, okay? The humans has like about 30. So among these millions of CGs, you have to find the needle in the haystack. You have to find those that they regress perfectly with age, either down methylation or hypomethylation at different levels. And this is the basis of epigenetic clock development. This was developed by this guy from UCLA in 2013. The paper was rejected from Nature Science because it was so good that nobody believed it. But finally, he got it published. And basically now, as you know, we have now a method. There is for companies now that can assess your biological age. That when people talk about biological age, now we know what we're talking about. We're talking about epigenetic clocks. So basically, you have like a age, the chronological age, the one in your DNA, and then you have like the epigenetic age. And then you can tell whether a person is like above or below, so it means that age is in a healthy manner or in an accelerated manner or not, or whatever. No? So this has prompted the development of many epigenetic clocks for mammals, no? And for birds. And the characteristic is that they are warm-blooded species. And then the question was like, what would happen for fishes? Fishes have two different or important characteristics compared to mammals. One is that they're cold-blooded, so therefore they are subjected to environmental influences. And the second one is that they have the indeterminate growth. So we have determinate growth, we grow until a certain height, and that's it, but the fish have indeterminate growth. So this was a challenge, but we managed in a very, I would say, like rough manner to develop the first epigenetic clock in fish. And this set the course, or this set the proof of concept for development of many different species. And now there is an avalanche of papers coming in the literature that start coming with the development of epigenetic clocks in fish for many species, for fisheries management, so forth. You know. And the way that these clocks are built, and this so far is not a problem, is that you start with the methylation data. In this case, the code, this is still not published. In methylation data, for example, in this case of the few millions of CPGs in the genome, then you use semi-targeted approaches in which you start with 47, in this case, 1,000 variables, okay, informative variables. That's a lot of variables. Typically, you use a sample size between 70 and 130 for epigenetic clock development. So then you split the data, 75 and 25, the typical thing. Then you do data preparation, exclude variables with 0 or near 0 variance, then you have less. And then you exclude variables with highly correlated between them. You put a threshold, then you get less. Exclude variables above and below a certain threshold, so forth. No? And then you end up with, you reduce this a lot, but still is a lot of variables to deal with. And then what you do is, like this 12,000 in this case, so here you have a matrix in which you have like 1, 2, 3, 4, 6, in this case until 12,000, and then you have the different ages, and then you have, what you do is you add a coefficient for the weight of one of these, each CPG's loci. And then what you do is use penalized regressions, which is a type of regression that has a compromise between like lowering the variance at the expense of increasing a little bit of the bias. And then there are three: the ridge, which many CPGs are put to the beta to coefficients to near 0 but not 0. And then the lasso, which is in a situation in where many CPGs are forced to have the coefficient to 0, so then you remove them from the model. And then the elastic net, which is a compromise between the two of them. Because of the complexity, nowadays ridge is less favored. So then, in this case, we use lasso and elastic net, and then the evaluation is the resampling, and this can be done by tenfold cross-validation or leave one out cross-validation, whatever. No? So then you build the model, and then you get the final model in which you have a very good accuracy. So, for example, this is the typical model that you do is like this, no? This is a regression line, is a typical linear regression. So you have estimated age is an intercept plus this lambda times like this equation. This is one part of the elastic net and this one part of the lasso. So the alpha value is 0 for ridge, 1 for lasso, and between 0 and 1 for elastic net. And lambda is the amount of shrinkage. So here you have the maximum absolute error and the root mean square error, and you compare the two models. They more or less give the same performance here during the testing of the models, but in this case we select lasso because the number of features, which in this case I should say is quite high, because normally in fish at the end in the epigenetic clocks in fish so far the number of features selected at the end is about 25 to 30, in this case it's quite high. But then you end up with something like that, okay? Something like that in which you have chronological age and predicted age. And this is something for the people who don't know about fisheries, something that is quite an advance because in fisheries you use otoliths, and the otoliths, which are these hard bone structures, they have a precision sometimes that between 1 and 2 years. And in many species, tropical species, some species, you cannot even use them because they are not reliable. No? So here you have like this method in which you can predict age, in the case of the cod, with 3 months of accuracy, which is pretty good. But so this is more or less solved. But so far this is the cod situation, no? Where you can develop the model because you have otoliths that you can use. So you have like the samples around 100, then you have the CPG sites, the variables that can be thousands or tens of thousands or even millions. And you have this type of matrix, and this will be an individual observation. So the chronological age here is known. But what happens with other species like hake, for example, in which the otoliths are not reliable? So here you have like different ways of assessing like the growth of this fish. And here you have the problems, and that's a current problem, that we have like, in this case, chronological age is well known for developing the model. In this case, the chronological age is not known. So the situation so far is use what's called projective inference, okay? And in this case, this is something I think that is pretty new. I don't know all the literature about that, but I think that is something pretty new, which is construct the best possible predictive model using a combination of all the features, although it uses a lot of features, but doesn't matter. And this is known as supervised statistical component analysis. You probably know much better than I do. And then the model is too complex, then you try to find a simple model in which gives a similar, the same type of similar predictions. And then for a given complexity, then the model with the smallest predictive discrepancy between the reference model and this model, then is the one that should be selected. But the important thing here, and the twist that we're trying to apply, is that we include the standard error of the response variable in the model where data are the supervised components. So here, going back, here we introduce this noise, this uncertainty in the model, okay? And this so far is the performance of the model. So the second problem is the use of epigenetic, the dealing with low sample sizes for machine learning-based methods application, and use of epigenetic biomarkers and their integration for selection programs in aquaculture. Here, what we ask is we can develop a strategy, combine machine learning procedures for starting using a small sample size. In aquaculture, you have nucleus of selection, what's called the nucleus of selection, that can be only of a few hundred fish. And to deal with them with these expensive techniques is complex and is hard working. So here, to summarize the workflow, is like we rank 59 families according to KPIs, key performance indicators like weight, survival, so forth, resistance to high temperature. And then we select 2 classes, the best and the worst families for that. And then we did this genome-wide approach at RDS. We have an initial population of about 500,000 CPGs to deal with, and then a statistical analysis of common DMCs in a sperm and offspring, 52 candidates, and then this going down to until we get 11 candidates. And then there were several rounds of validation. I'm going to explain this in overview. I'm going to explain this now. So then we use different next families, and then a targeted approach, still a statistical validation, second validation only with the sperm samples because we wanted to use a marker that could be inheritable, so therefore we have to make sure that it's present in the gametes. And then finally an external validation with the sperm from completely unrelated families. So here we start with the overall DNA methylation differences between eggs, ovaries, testes, and sperm. And then we selected 52 differential methylated cytosines that are common between the sperm and the gonads, so these 2. And then applying different machine learning models, we end up with 11 CPGs. The most voted are these ones, but after statistical testing, we saw that one of the most important ones was not among these 3, so we put these 3, and then we selected finally the 13, the 31, and the 30, the second best one with the models. So this is validation 1 and 2 using the support vector machine algorithm. So batch 1, 20 samples are RDS. This is definitely an overfitting of the model. That's why it gives this value, only small sample sizes, with the 3 CPGs initially. Then batch 2, 70 samples with a targeted approach. Then this is not validated because it's not better than tossing a coin. So this is not a validation. And then we change the 3 initial CPGs with these updated ones. Again, this works, but probably is overfitting. Then we try with 70 samples like here, MBS, the 3 new ones. Then this is validation 1. And then different type of samples, batch 1, 3 markers, validation 2. And then batch 1 and 2, but sperm only, the 3, and then there is also a validation. And then finally, the external validation is using a sperm from a different group of fish. So first, we predict the class of the sires based on the model built. So again, we look at the sires based on the model that we built with the training set of batches 1 and 2. And then we have like external batch samples, only 8, that's what we had. The same 3 markers, and then we check the phenotype data regarding the KPIs. And here we have like we have here the biomass, which is what we look for, the class by phenotype, what we say best and worst, and then the model how it classifies. And 7 out of 8 samples were correctly classified. But again, this is perhaps like pushing too much the system, but this is the best thing that can be done. And I think that is a way to overcome the fact that we have to start with small sample sizes because of the type of things that we deal with. So, and the last one is the proper estimation of a bound of epigenetic inheritance. And this is, again, is a very hot topic. There is a journal developed on environmental epigenetics with a special issue on that. And we want to look at the epigenetic inheritance to improve aquatic production and to assess how organisms will deal with climate change. This is something that for many people has been confused with Lamarckism. I have a slide, an extra slide at the end if you want to see, but it's not Lamarckism at all, right? So have this clear. And here there are 3 different approaches to try to assess epigenetic inheritance. And again, here is not only a mathematical problem, but also it is a problem of concept. No? Like we still don't know exactly how to measure, and there are different ways of measuring things. So one simple thing is to measure like overlaps. So if we have one mark in one generation and this mark is in the next generation, then we can say, okay, this has been transmitted to the next generation. But hold on, because this can be a problematic. Another thing is like a simple regression, parents methylation, offspring methylation. And third one is a stable CPGs across generations related with what's called methylation quantitative trait loci. So again, there are different methods, and here, for example, you have the grandparent, like the 2 childs, and then the parent, then the offspring. and then you have the offspring and the uncle. No? So in each case, there is chances for resetting this epigenetic information, and this is something that needs also to be taken into account, and this is not properly taken into account. So the studies so far, very few. In mammals and birds and fish, we have average sample sizes of 88 in a range between 24 and 238. Our studies, 95 and 130 samples, are within what is currently being done. And what we did is see what happens when you expose animals to elevated temperature, for example, and then what happens in the next generations, not only in the exposed generation, but in the next generation. So you have a situation where you don't have exposure, so the control, then during development, the ancestral exposure, and then development and ancestral. And we want to see, in this case, is all these epigenetic marks that are induced by the change in the environment, how they are transmitted in the unexposed offspring, all right? And for this, we did 3 tests, stable CPGs. So here we're comparing these ones with these ones and these ones with these ones. And what we can see, so the ones that are stable, no DMCs between the F0 and the F1 is about 85%. But of course, this can be simply due by chance. And therefore, you have to apply permutation. So in this case, we compare the F0 untreated and the F0 treated. We get about 10,000 DMCs. This is about 2% of all the captured DMCs. And then also the same thing in the offspring. So we see that the types of overlap. And what you can see roughly is that, it depends on the sex, but the values are very similar, 4 to 5, that the environmentally induced in the F0 that are present here in the unexposed F1 is about 5%. And this is a value that more or less matches with what is in the literature. Between 0 and 10, 15% maximum of the CPGs induced by the environment can be passed to the next generation. And then here, permutation. So here we're working still, not getting the results that we wanted, but this is the type of results or the type of studies that we're doing. So this is a permutation test called methyl inheritance based on Monte Carlo algorithm. So the take-home messages is that, like in biomedical research, many of the traits of interest in food production and biodiversity conservation in the face of climate change are the result of complex genetic interactions and of polygenic nature. So this is like bad news, but this is something that we have to learn to live with. No? That epigenetic integrates genomic and environmental information and has a key role of shaping the organismal phenotype. No? And we know that this heritability, missing heritability problem can be explained quite a lot for epigenetics, but we still need to develop further tools to how to assess this. The environmentally induced epigenetic changes have the potential to be inherited. The fact that in aquatic organism development, fertilization and early development is external, this poses opportunities and challenges. Opportunities because we can manipulate this challenge. So this is good in the terms of like aquatic production or aquaculture production, but this can be bad for wild populations in the face of climate change. And then epigenetic biomarkers have some advantages. They can be like not only diagnostic but also prognostic value. But we need solutions to these 3 problems that I mentioned. No? So especially to deal with the contribution, how to get rid of many of the predictor variables that not contribute enough, how to find the needle in the haystack, dealing also with low sample sizes for machine learning, because sample sizes are not that easy to get, especially in a small and medium and small companies, and proper estimation of the amount of epigenetic inheritance. All right, so thank you very much.
Nice, thank you, fascinating. We have 10 minutes of questions. Any question? Nobody?
Everything clear.
Okay, so we can break for the coffee break. We can slowly move to the entrance, no, Sonia? In the entrance of the institute, there's a coffee break and there's a poster, so don't forget to look at the posters. Thank you.
Thank you.
to leave your stuff here or we close?
Aquí tenim el Pere Puig del CRM. Ah! És que clar, no tenim Pere Puig. Jo també ho vaig veure, però dues vegades.
I li he comentat que ens coneixem.
En sèrio?
Ens coneixem perquè la Maria va ser endavant i coneixem el Marta.
Però em vaig entendre o formació del...
Sí,
Pues ja estic
fa molts
ubicat,
anys això, fa molts anys. Sí, sí, i
Bueno, clar, és que jo ho vaig tenir que mirar dues vegades, quan ho vaig veure, dic, ostres. Jo no posava això, però sí, és el que no va dir, falten les presentacions després. Sí, sí, també.
a mi em toca a les 11, no?
Sí. How many dolphins are in...
Vale. Mira, s'ha de posar aquí. Sí, i em faltarà l'altre aquí.
No, no està damunt. Sí, no sé.
On la poso? La carpeta aquesta que has obert?
Sí, mira, perdona.
Aquesta? Bueno, l'he posada en el...
Està aquí? Doncs aquí mateix, ja està.
Deixa'm obrir-la i veure si a
tot.
les fonts i tot això funciona. Es veu bé.
I aquí tens per passar, que la gent no ha fet servir, però això en teoria...
Aquí també és un làser.
I és làser... Sí. Val? Per passar endavant i enrere i el làser el tens aquí.
Bueno, en principi sí, tot va bé.
Fantàstic. Doncs la deixem... Ja si vols la deixem...
La deixem posada?
Sí, no? La primera slide, si vols.
Com vulgueu.
Sí, i així anirem tot més ràpid. Vale, i et deixo el...
llapis.
Vale, fantàstic.
Molt bé.
Sí, ara tanquem la sala.
Why this title? How many fishes are in Sepetiba Bay? Well, my intention is to explain you something about capture-recapture methods, okay? And this is only an example, the number of dolphins in Sepetiba Bay. Look, these classical titles concerning simple capture-recapture models, the estimation of total fish population of a lake, the estimation of fish in lakes and ponds, and so, okay? And then, first of all, let me explain you some very simple idea on capture-recapture models. Okay, how many fishes are in this pond? How to count them? Okay, of course, we cannot dry the pond and count the fishes. This is ecologically unacceptable, right? Then, a way for counting the fishes could be the following. First, we capture a group of individuals alive, okay? And we mark them in some way, okay? A mark could be also a picture, for instance, if you can identify the fishes by a picture. For instance, if you consider the Osmerus of Meda Island, they have some marks, and you can identify the Osmerus using pictures, for instance, right? Then you take some of them, and then after that, you put again in the pond and wait some time, and what happened? They look that are distributed more or less uniformly in the pond, right? And then this is the first capture. The first capture we have done is 5 fishes, remember. Now we perform a second capture, another sample of individuals. And what we see here, we see that some of them are unmarked, but we find 2 previously marked, right? Then this is a recapture. This is the idea, capture, recapture, okay? And then how this information allows to estimate the number of fishes in the pond. How? Consider, let N1 the number of individuals in the first capture. Let N, capital N, the unknown number of fishes in the pond. What is N1 divided by N? This is the proportion of marked fishes, right? And this proportion has to be approximately equal to the proportion of marked fishes in the second capture. M is the number of individuals that are found marked, and N2 is the sample size of the second capture, right? Then for this very simple relation, we can estimate capital N, the number of fishes in the pond. It's very simple, right? Perhaps many of you know that this is known as the Lincoln-Petersen estimator of the population size. Okay? Well, for our current example, 5 fishes captured in the first sample, 8 in the second, and 2 found marked in the second. It would be the estimated number of fishes in our toy example. It's very simple, right? It's very simple. Then, this is the most simple model in the history of capture-recapture methods. It's very simple. But capture-recapture methods is a very large world. There are many papers, many books concerning capture-recapture methods, many things. Look that this estimator does not have sense when m is equal 0. Okay? And then this estimator is biased and does not work well for values close to 0. If m is equal 1, for instance, or 2, this estimator does not work well. Then, usually this estimator is used in a modified form due to Chapman in 1951. This modification of Lincoln-Petersen estimator is much better and is more unbiased and allows to estimate samples, population sizes when m is equal 0, for instance. What the mathematics behind this? Because look that this is a very simple relationship, no mathematics are needed for deducing this. But the study of these estimators are based on that fixing the sample size of the captures, N1 and N2. What is random here? Here is random the number of fishes found marked in the second sample. Well, it follows a hypergeometric distribution. This is the probability function for a hypergeometric distribution and so on. Okay? Well, of course, we can evaluate also uncertainty. How? Computing the variance of the estimator, and also we can calculate approximate confidence intervals. This is a classical expression for doing that. Something about the history of this estimator. Remember, the name is Lincoln-Petersen estimator. This is George Johannes Petersen that used this method at the end of 19th century. I think that for estimating the population size of salmons, right? In 19th century. And Frederick C. Lincoln used this method for estimating the populations of birds, especially ducks, in the United States in the 30s of the past century. Well, these are the popularizers, but not the inventors of the method. Because this method is very old, is really very old. And the first time that capture-recapture is used, and this estimator is used, is due to Laplace, the mathematician Laplace. Laplace used this estimator for estimating the population size of France at the end of 18th century. But how here for people capture-recapture, what is the meaning of that? The meaning is that in this case an incomplete census was the first capture, the people in an incomplete census. It was just N1. And then the Laplace team also performed a survey along all France, and the people captured in this survey were the second capture, N2. In this way, Laplace estimated the population size of France at the end of 18th century, right? Well, it's important to mention that this method not always works. And this is the model assumptions. The population to be studied has to be closed. It means that there are not births or deaths during the period of study. But samples have to be random samples. All the individuals have the same probability to be captured. For instance, a big fish and a small fish have to be the same probability to be captured. Not always happens, right? Capture and marking don't change the probability to be captured. For instance, suppose that the first capture is very aggressive, and when the fishes, the second capture, look at the net or something like this, go away. This is not good, right? Okay? And of course, no marks fall off individuals between captures. These are the assumptions of the model, right? Then, well, in social sciences, capture-recapture methods are frequently used. For instance, this is used for estimating the size of populations that are called hidden populations. Hidden populations could be, for instance, drug users, or could be women who have been mistreated, or homeless individuals, and so on, right? It's very used, this methodology, in social sciences and also in health sciences for counting the number of infected individuals for some diseases and so on. Moreover, sometimes Lincoln-Peterson estimator is rediscovered, and someone says, well, I've invented a method for doing that, or something like this. This is the case of this paper in 2001 that is a model for estimating the number of near-Earth asteroids, that is, objects near to the Earth that can be dangerous for people, right? And then the authors of this paper use a Lincoln-Peterson estimator, and they probably thought that it was new, absolutely new, right? Okay, then this is, as I mentioned, this is a very simple model. The model can be complicated, and for instance, look, this paper published in the International Journal of Science and Society in 2011, using capture-recapture to estimate the prevalence of intimate partner violence. These are here the authors. What they use? The authors here use a more complex capture-recapture pattern. Until now, or the Lincoln-Peterson estimator is used with two lists, two captures, right? Here we have three captures. One is the women that are in the list of the prosecutor's office. The other list is some shelters for local domestic violence. And the other list is the family outreach agencies. And here you can see the numbers. For instance, 155 corresponds to those which are in the list of prosecutor office, and they are not in the other list, right? In the intersection, you can see the repeated person in several lists, right? Okay. Well, this is a model that shows the complexity of capture-recapture method. For estimating here the population size, it's not so easy, like Lincoln-Peterson estimator. And here we have to use, for instance, log linear models for doing that, another technique. Well, then, let's go now to how to estimate the number of dolphins in Sepetiba Bay. Dolphins can be identified by their marks in the fin. Look the marks, and look here the marks. This is the same fin, right? And well, there is a software for photo identification called Darwin software that the authors of this paper have used for estimating the number of dolphins. And this is Sepetiba Bay in Rio de Janeiro state, here in Brazil. And this is the papers where they estimate the number of dolphins here, right? And look, the title, Capture, Recapture, Abundance Estimate of Guyana Dolphins, and so on, right? This is the paper. And let's go to see the methodology. And after that, I'm going to explain what is new, what is our contribution to this methodology. Here, this is the final result of the research in this paper. What is the meaning of this? 228 dolphins have been sighted just one time. A sight here is a capture. Right? 110 have been seen 2 times. 19 have been seen 3 times, and so. And 1 has been seen 6 times. This is the most sociable dolphin, right? Okay? Then, in total, we have observed 382. And this is a multiple capture-recapture experiment. And then, our interest is to estimate the number of zeros. That is, how many dolphins have not been observed, right? Then, mathematically speaking, we have here a count distribution. This is a count distribution, truncated at 0. This is the technical words, if you like. Truncated at 0, and our interest is to estimate the number of zeros. This is a mathematical problem. Right? Then, how to do this, and how people in some sense solve this problem. This is very similar to the following problem. How many words did Shakespeare know? Right? And this is the number of words that, for instance, how many words Shakespeare only used 1 time in all his works, 40,376. How many words Shakespeare only used 2 times in all his works, and so. Okay? And then, looking this table, we can ask about how many words did Shakespeare know but never wrote, right? This is an equivalent problem. And this problem was solved, or partially solved, but Bradley Efron, for instance, and other famous statisticians. And in total, Shakespeare wrote around 1,000 different words, okay, in all his works. But how many did know, right? Well, then let's go to see how we can estimate the number of zeros. Look that this sample corresponds to a zero truncated distribution where the estimated probability of 1 is just 228 divided by 382, this number, and here we have the estimated probabilities of the truncated distribution, okay? Then, this is sum of mathematics, right? Given a count random variable, these are the probabilities, the probabilities of the random variable truncated at 0, that would be denoted in this way, x plus, means that we have excluded the 0, right? These are the probabilities. Okay, these are pk divided by 1 minus p0. And then, how to estimate the number of zeros? First, let's go to explain parametric estimation. What means parametric estimation in statistics? It means that we assume a known distribution for the data. Okay, for instance, Poisson. But normally, we don't know the distribution of the data. Then it will be non-parametric estimation when we don't know the distribution. But now we assume that we know the distribution. Then, because we know the distribution, this is the probability function, and then the parameter of the Poisson can be estimated by classical methods in statistics like maximum likelihood, and then the proportion of zeros can be estimated in this way, that is exponential minus the estimation of lambda, because this is the proportion of zeros for a Poisson. No problem. This is a classical, absolutely classical statistics. Finally, the population size is estimating using the so-called Horvitz-Thompson estimator, that this is this expression where n is the number of dolphins that have been observed, that is 382, and this is the estimate of the proportion of zeros. This is parametric estimation. But you have to assume that the distribution of data is known. Of course, you can see, well, there are other distributions. Yes, instead of Poisson, you can take, for instance, negative binomial, this kind of distribution, right? But anyway, we never will be sure that the distribution is correct, is right. Okay, then, non-parametric estimation. Bad news. In general, given the distribution of the zero truncated version, in general, it's impossible to estimate the probability of zero. It's impossible, in general, given an arbitrary truncated distribution. It's mathematically impossible, right? Then, anyway, probably you know the Challanges estimator that is considered non-parametric, right? How it works. Challanges estimator that was invented by Ann Chow in the 80s assume that the truncated data, comes from a mixed Poisson distribution. What is a mixed Poisson distribution? We say that a variable follows a mixed Poisson distribution when x follows a Poisson, where the parameter lambda is also a random variable. It doesn't matter which kind of random variable. Could be uniform, could be, it doesn't matter, right? Then all these variables are called mixed Poisson distributions. And Chow, 1984, 1987, found a very useful inequality that, with this inequality, she established the Challanges estimator, the so-called Challanges estimator. That is, the proportion of zero for any mixed Poisson distribution is always greater or equal than the proportion of 1s to the square divided by twice the proportion of 2s. Okay? This is the classical Chow estimator. Then, Chow's estimator is the following. The Chow's estimator is equal to the number of observed dolphins plus the number of observed dolphins to the square divided by twice the number of observed dolphins 2 times. In other words, well, this is very simple. But anyway, because this estimator comes from an inequality, this estimator provides a lower bound of the population size. Okay? And is considered non-parametric because it works for all mixed Poisson distribution. Right? For dolphins data, applying to dolphins data, we find this value for the estimate, and we also can compute a confidence interval, and this is the result, right? Well, this is Anne Chow. These are the papers where she introduced the Chow's estimator, that is the gold standard in non-parametric capture-recapture methods. Look, the number of citations is really high, okay? And in 2017, I was the editor of this journal, Statistics and Operation Research Transaction, and I invite Anne Chow to write a paper concerning the contributions during these 30 years, right? And you can download this paper from this website because it's open access. And here you will find, well, many things, many topics related with Chow's estimator. Then, now let me explain you our contributions about this. First, some math. What is the probability generating function, that is PGF, of a random variable? This is nothing more than a power series of the probabilities, right? The probability of 0 plus the probability of 1 multiplied by s, probability of 2 multiplied by 2s squared, and so it's a power series with the probability. This is a PGF, no? Then, the PGF is very important in probability theory because the derivative of the PGF at s equals 0 provides the probabilities. Moreover, the derivatives at s equals 1 provides the moments or a linear combination of the moments. Then it's very useful because with only one function we can describe many properties of the distribution and so on, right? Any random variable is absolutely determined by its PGF. And for instance, for Poisson distribution, this is just the PGF. Right? This is just an example. Then, remember mixed Poisson distributions, remember the result of Chow. This is a very large set of random variables, those such that the logarithm of the PGF is a convex function. Yes, a convex function. Okay? What is this? What is the meaning of this? Is this a large family? Yes, this is a monster. Okay? And then let me tell you some properties of this class of random variables. This class contains all mixed Poisson distributions. Then the result of Chow can be improved. Contains also another family of distributions known as compound Poisson. contain distributions that are not mixed or compound Poisson. Different, absolutely different. Nothing related with the two previous families of distributions. Independent sums of members of this class also belong to this class. Then if we sum random variables, we are inside the class again. And finite mixtures of members of this class also belong to the LC class. Then with all these properties, we can say that is really a very large family of distributions. You can read the details of this family of distributions in this paper published in 2018. If some of you are interested in this, please write me and I can send you the paper and so. Okay? And then, let me show you the power of this class of random variables. Suppose that we have a random variable such that the probability function have this form. Okay? Then, is this random variable mixed Poisson distributed? Is compound Poisson distributed? This is very difficult to answer. Probably, well, I don't know how to answer to these two questions, but I know how to answer to the third question. Does this random variable belong to the LC class? It's very simple because I have to plot only the PGF numerically, and yes, is convex, then belongs to the LC class. It's very simple. Right? Then LC class is very powerful, right? Then our method of estimation. Now, we forget mixed Poisson models because are included in LC class, and then we assume that our counts belong to the LC class, to any of them. Then we have found the following inequality, where it indicates the derivative of the PGF of the truncated distribution, and this is the second derivative of the PGF of the truncated distribution. Then this is a very powerful inequality because when S is equal 0, it leads to Chau's inequality. But S can be any value between 0 and 1. Okay? Then this result, only this result, improved the previous result of Chau. Then the sharpest inequality corresponds to computing the maximum value of this function. The maximum value, the S for which the maximum value is attained, is called S star. And finally, because this is the best inequality of all them, we obtain this inequality where C is this function evaluated at the maximum. Okay, then working with empirical PGF, we obtain the following lower bound, and finally this estimate of the population size. Then let me show you how it works. Okay? Coming back to dolphins data, this is the estimated probabilities, 1, 2, 3, 4, 5, 6, that's each number divided by 382, very easy. And this is the empirical PGF. Remember what is the PGF? The PGF is a power series with the probabilities, okay? Then the probability of 1 is here, multiplied by S. The probability of 2 is here, multiplied by S to the square, and so on. This is the empirical PCF. Okay? Now, remember, we have to compute this function and to find the maximum. Then, well, this is the function, and now we have to compute the maximum of this function. How? Of course, numerically, right? With a program that we have and is available for you. Okay, if you write it. And this is the function, and look that we have a maximum for s equals 0.24 approximately, that is here. And then the estimated proportion of zeros is this one, 39.5% of zeros. And this is the population size, the estimated population size. Right? We can also compute a bootstrap confidence interval. Okay? Well, this is our method, and we are working in variation of this kind of things. But the final message, in some sense, think that capture-recapture can be used not only for estimating number of animals or species, but it can be used for other things, right? And this is a joint work with María Dolores Jiménez Gamero from Sevilla. It has been published in the Biomedical Journal. And moreover, we have also details and more examples in chapter 11 of this book. Okay? That I'm the author of this chapter. And thank you for your attention.
Thank you very much. Very interesting question.
Thank you very much. Now, I have a question on the spatial space variable in this problem. I mean, if the domain is very large, and then you are capturing
the space of,
yeah, I mean, it's not a bay, but the whole ocean, for instance. How does it affect on the capturing and recapturing zones of distribution in the space?
In the space?
Yeah, I mean, yes. How do you have to do the capturing and recapturing?
Marking the place where have been taken the capture? Or
Yeah, if you are working in the whole ocean, and then you do capturing or recapturing in small areas, how can you? Yeah, how can you deal with that? Or if you have to do it in the whole thing, or how do you manage with this uncertainty?
This is not easy, but this is a problem where there is not homogeneity. Probably it's a problem with heterogeneity. Okay, and then well, the problem has to be studied for each case. But a way for doing that is, for instance, using a stratification. If you can consider several areas and analyze separately, and then joining all the results, it would be a method, for instance. You can also analyze the capture-recaptures, taking into account the time, like a variable in the time, and also a variable could be a spatial variable. You can take into account the specific places, right? The model, depending on the species or depending on the animal or so, have to be studied indeed for providing a good answer to your question, I think. Okay.
Hello, thank you for your talk. I wanted to ask if, I don't know if this thing exists in Earth, an area where you are 100% sure about the number of species, and if you have tried this estimation of how many species, knowing already the answer, but have you tried this methodology observing this number of times as you see dolphin, or to estimate to test the validity of this approach?
Yes, your question is related that if this methodology can be used for estimating the abundance of a species in general, yes? Well, look that, in fact, the answer is yes, but let me show you. Well, in this paper, Chow was mainly interested in estimating the number of species, and this is one of the topics that can be analyzed using capture-recapture method, right? And for instance, one of the last papers of Chow is related and how to estimate the number of species in reef coral, in an area with a reef coral, using citizen observations. Using citizen observations and counting she used only two kinds of data. Those species that has been seen one time and those seen more than one time. Yes? If you are interested, I can send you the paper.
More questions?
Okay. Is there any important example of a distribution which is not in the LC class?
Yes, good question, good question. Which distributions are not in the LC class? For instance, those that are under-dispersed. What is the meaning? Over-dispersion for a count distribution is when the variance is greater than the mean. This is the case, for instance, for mixed Poisson distributions, for negative binomial distribution, this is a special case, and so on. And under-dispersion is when the variance is lower than the mean. Those distributions are not included in the LC class.
It's a bit off, but I would do an experiment. I mean, we can do experiments in these things. I mean, not with the dolphins, right? But with balls in a buck. So you can actually test whether the estimates are good or not, and they fit the model better than other, or things like that. So is that something that people do, or?
Yes. Well, there are some papers where there are real experiments where the number of animals is really known, and then the methods, the capture-recapture methods, are tested in order to check if they work or not. No? There are papers about this. And another way to make an experiment is, of course, by simulation. And, well, in all papers we have performed some simulations and they work well, and yes. Yes, the answer is yes, but of course the more interesting experiments are the real experiments. For instance, there is one famous experiment in capture-recapture concerning, and I think that Chow explained this in one of her papers, about the number of taxis in Edinburgh. You know the number of taxis in Edinburgh, and you also perform a capture-recapture method for counting them. That is, observing and recording the license of the taxi, right? And there are some examples of this kind.
Yeah, but these type of experiments, I mean, or the natural experiments, or the one with the dolphins, that are subject to random effects, to all sorts of things that might happen. No? I mean, a taxi driver stops and goes for a coffee, and things like that, right? So to me, it would be, and this, I think this reflects also the way of thinking about the problem. No? I would be tempted to do the experiment with the labeled balls in a buck and try again and again and again and see whether I get an approximation to the number or to get insight into the rules that are behind a specific problem. And it's an approach that's completely different to the one you took, which of course it comes from your background and my background, right? So I found that funny anyway.
More questions?
Hi. Okay, so thank you for the talk. I think I've learned more statistics today than in my undergrad studies in biology, so thank you. And I have a question. So have you applied these techniques to other data sets? Because I think you did some work on COVID-infected patients. Can you talk a little bit about it?
Well, well, this is one of my topics of research, but as you mentioned, it's not the only one, right? And concerning COVID, I've worked for predictive models in COVID and so, but using time series techniques, not capture techniques, right? But there are some papers related with the use of capture-recapture techniques for counting some kind of, for instance, sexual transmission diseases and these kind of things. Diseases that are not easy to control or to know what happens.
Another question?
Hi, thank you for the talk again. In geoscience and in geohazard assessment, it's quite important to understand frequency magnitude distributions. And frequency and magnitude is time and magnitude to axis. So basically, is there a way to estimate a population divided by the different magnitudes within the population? Let's say, for example, sizes of fishes with respect to number of occurrences, let's say. To understand the population size is divided by the size of each population, by each population range.
Well, probably yes. I'm not sure because as I've commented you previously, the world in capture-recapture methods is very wide. And if you are interested in a specific problem, I can help you to look for a specific solution and to work in the topic. And so probably the answer is yes, but I'm not absolutely sure.
I'm saying this because usually the larger events are those that are better constrained, but as you move to smaller and smaller events, you have more uncertainties on the size of the actual population of those events.
Thank you for the talk. I'm really bad in math, but I've been using the method of Anne Chow to estimate species diversity based on implicant sequencing, so microbial diversity in our samples. And basically my question relates to the nature of the data and how it fits with the method or not, because our data in sequencing, it's compositional, and I wonder whether I've been applying this thing in the right way or not. Are we allowed to do this?
Your data is compositional. Your data is compositional. That means that the proportions have to sum 1, right? And well, I don't know if capture-recapture methods can be used in this way. But anyway, using compositional data methods, there are many options for working with compositions. There is a strong group in Girona, but I don't know if you know them, working in compositional data analysis. That the principal investigator is or was Pere Puig, actually. They have a large research and a lot of papers on compositional data analysis and okay.
More questions? Otherwise, I have a question myself. So here in fisheries science, we work, so when we capture our organisms, they all have a catchability coefficient. So yes. It's not the same, the same probability. Do you know if these methods have been adapted to, I know that the assumption of the model would be violated, but is there any adaptation of the method to?
Yes. There are many research and many papers using ideas on catchability and the effort also of the catchability, and all can be combined for estimating the population size. Population size can be estimated not only using capture-recapture methods, but with a combination of methods. Estimating catchability, estimating the effort for doing that, and with all this information can be plugged into and to get a reliable method of estimation, or a reliable estimate.
Nice. Any additional questions? Otherwise, thank you very much, Pere.
Thank you.
In 15 minutes, we have the lunch break. You are all invited to have lunch with us in the same place where we had the coffee break. And we have some minutes for networking, slowly making it to the entrance and keep discussing. Thank you. De la tarde. Yeah, so basically after the lunch, we have 2 rooms booked in case some of you want to discuss together some projects or some issues. Just let us know and we'll open them for you, okay? Otherwise, we'll see you all tomorrow at 9 for the next talks.